So, bonjour, good morning, everybody. Welcome back to our Physics of Medical Imaging class. I'm just checking, as always, that everything runs. And everything works. Well, today's plan you already can see. Today our goals will be less ambitious. So we are going just to learn something about the advantages of fluorescence microscopy. And in doing so, we are going to check some common fluorophores used in fluorescence microscopy. We will get certain knowledge about the light sources typical for fluorescence microscopy. And probably we will learn some terms related to filters and mirrors, like dichroic mirror, like pass filter, band filter. And the other two terms we are going to talk about are Jablonski diagram and Stokes shift. Well, this is the program for today, and it's gonna be not hard for us. So as compared to all other models we had before, that is definitely easier to follow. First of all, let me give a very short recapitulation of what we've done previously last week. So we already overviewed some features of microscopy and some limitations that are well, already sort of inborn in microscopic systems. One of them was related to the wave nature of light and the fact that optical system mostly cuts part of the waves. And last week we've learned that optical system usually cuts the waves that carry high frequency spatial information, which means that any attempt to decrease the opening of optical system will lead to compromise or to worsening of resolution power. And in order to illustrate that, I use the concepts of wave numbers and K-space. Today, we will begin with so-called Louis de Broglie equation, which will illustrate some important compromise, or we can call it trade-off, that need to be made in cell imaging. Let's go ahead. The formula I'm talking about, many of you probably already know, and it's very easy to remember it if you need, due to its simplicity. Uh, as you know, there were many people who paved the way for quantum physical understanding of the world. Among them were such people as Max Planck, who actually said that nature is quantized, energy is quantized. Albert Einstein, who said that energy and matter are in fact very much connected to each other, so we can always consider matter as a form of energy and vice versa. And also Einstein illustrated how this relationship can be calculated. But one very fundamental, very influential idea in the quantum physics was the fact that every material object in the world is at the same time a wave. And this is what they called material waves or matter waves. And this idea comes from Louis de Broglie, who was getting, I guess, financial education, and then luckily decided to proceed in physics. Well, the equation which illustrates the wave nature of any object is shown here. That means if we have some object having momentum of motion p, as you may know, momentum of motion is just a product of mass and speed or velocity. Mass and velocity multiplied, mv. In German, we call it impulse. So as if we check just impulse of this momentum of motion of this object, and if we divide the Planck constant by this momentum of motion, we will get certain value, and this value is going to represent the wave length typical for this object. That means we can do this calculation for me and for you, for example. At the moment, I'm riding my bicycle. I have certain momentum of motion, and I can calculate my own wave, which is intrinsic to me. And the same can be done for any material object, including, of course, some elementary particles. As is also obvious from this equation, the tiny the particles are, the smaller they are, the larger will be the wavelength. Or I should say it better, the smaller the momentum of motion of the particle is, the larger will be the corresponding wavelength. And this brings us to some important practical conclusion. That means we wanted to have shorter wavelengths. We know that the shorter the wavelength, the better is actually resolution of your system, of your microscopic system. So that's why we said that blue light or violet light in optical microscopy is actually better. And if we go even shorter, we get chance to catch more details. So the shorter the wavelength, the better the resolution in general. And that means if we use, instead of photons that possess relatively small momentum of motion, if we take some more massive particles who have larger momentum of motion, we have a chance to produce shorter wavelength. So this is just the idea behind. So instead of using photons, we can consider taking some other particles that have larger momentum and that bring more resolution due to shorter wavelength. Okay, what particles that come to our mind? Obviously, the first thing we can think about would be electrons, and then next protons, neutrons, and some other particles. So actually, it can go further. And indeed, this idea has been realized in electron microscopy, as you know, and of course, most of you know that electron microscopy indeed brings much higher resolution as compared to the photon-based systems. And here the equation describes, explains why electron microscopy is better. But I announced this chapter before as a trade-off, as compromise. So what do I mean with this compromise? It seems to be nice. It seems to be no compromise. So you just take more massive particles and you get better resolution and you win. Perfect. Well, it's not so simple, and the main problem in using more massive particles for probing your samples is that it is getting quickly destructive. So this is the key word. At the moment you start, seeing your system or in checking it, examining it, you destroy your system. And that's getting a problem. As long as you use photons, it's actually sometimes destructive. We will see it later, but not so much. In fact, if you use electrons, you see it within seconds that your sample you are studying is just get destructed by this electron beam. And this is a problem, you see? So in other words, you have to find some good solution. You can use more massive particles, but you should do it very carefully so that you get the advantages of higher resolution, but you don't experience disadvantages that are related to destruction of your sample. How can it be done then? And now I would like to tell you something that probably will expand your knowledge about electron microscopy. If you know how electron microscopy works, maybe today I would be able to surprise you a little bit. Let us check the setup of an electron microscope. And by the way, here in Jülich, we have a very powerful electron microscopy system in the laboratory from Professor Schöning. And it's really a nice machine. So they allowed us also friendly to use it from time to time. Here was some typical electron setup. What they use, actually, it's mostly scanning electron microscopy system, but here we will consider the transmission electron microscopy. So we need some source of electrons, and this emitter of electrons produces some flow of electrons. It will be then focused by magnets, and we call it like sort of lens, electron lens. And then it goes through your sample, which is situated here, and later it will produce some image which can be then recorded by using, for example, a CCD camera or whatever. Well, this is actually some standard idea about how electron microscopy works. But now the new update, and this may be new for you. As we already discussed today, if you used an electron beam, that will be destructive for your sample. How can you then make it in a more gentle way, in a more mild way? And the solution was pretty funny. Modern electron microscopies sometimes emit electrons really one by one. So in fact, they do not produce really a flow of electrons that just goes through a sample. Some electrons get absorbed and some other electrons, they go through and you get an image. No, this idea is not true anymore. It's very much destructive. Instead of this, many modern microscopy systems work in a more advanced, in a more fascinating way. One single electron is produced by electron gun, goes through your system and goes through your sample and produces already an image, which will be not very bright. It will be quite pale, in fact. But the fascinating feature here is one single electron can produce an image. And then you say, wait a moment, how comes it just goes through one part of my sample? So here's my sample, and if I have one single electron, it just goes through, and I have no idea what the other regions they look like. It's just one point. But the truth is that here exactly where de Broglie relation appears, where it comes true. Emitted electron behaves as a wave. It has some intrinsic wave property. And at the moment it goes through your sample, it interacts with your sample as a wave. That means it goes somehow through all points of your sample. Looks quite crazy for people who are far from quantum physics, but you have one single electron. This is your sample you want to study, and as it goes through it, it interacts with this point, this point, this point, this point, this point, this point, all points of your sample, but it interacts with them as a wave. So it goes like this, one single electron. It produces some interferences where it can interfere with itself. Quite crazy enough, right? And so that means, again, we are in the range or in the domain where optical or wave properties will play an important role. And again, our previous considerations that we have wave phenomena and wave limitations, they also apply in our system as well here. So my main message to you is that electron microscopy has also many features of wave optics. Electron acts as a wave there and produces interference pattern, and like an optical system, meanwhile on the way here we have this Fourier domain information which will be then later due to interference converted into visible image. Okay, so at least to me, as I heard about this some years ago, it really sounded quite crazy, but this is how modern transmission electron microscopy works. So it's really wave-related method where electrons are emitted one by one. Since we will not take this topic too much deep in the scope, I think this is more or less enough what we should know about electron microscopy. Let me just conclude in showing some couple of more pictures. I'm a big fan of transmission electron microscopy because most of information we know nowadays about the fine cell structure, about viruses and many other events, it has been obtained by using electron microscopy. Here I just brought some images that you may find quite interesting. Here we have thylakoid structures in chloroplasts of plants, and this is where the photosynthetic processes take place. Here we have fine structure of our nucleus, including nucleolus, where we can see, even if we're lucky, we have single chromosomes to see. We can also see various inclusions, and even here, number 8, we see how virus particle, it is a virus entering the cell. Exactly the event when virus interacts with the cell, and we can even see which receptors it can use. So really fantastic possibilities. can open if you use transmission electron microscopy. So here we also have some limitations. That means mostly we cannot see single proteins. That means the resolution is at about 10 nanometer. All structures that are less than 10 nanometer, and most proteins are less than 10 nanometer, you cannot really clearly see. They will appear again smeared due to diffraction, due to wave limitations. But things like ribosomes, mitochondria, virus particles that are all in the size range between, let's say, 50 and 200 nanometer, they will be look really perfectly so that you can see lots of details. So now let me come back to our initial plan. This first point was to illustrate that electron microscopy uses, well, you can say explicitly or implicitly, the Broglie equation, and it brings with itself an advantage and disadvantage, right? So, and I think if we make it an exam question, an easy one, I would ask about what kind of trade-off you meet in electron microscopy, and then they say, okay, on the one hand, it's high resolution power, but it's very destructive. Yeah, so the question arrived, how do you control to emit just one electron? Good question. Yes, so it's indeed, this is what makes such systems quite expensive. Physically, or seeing from engineering point of view, they are not much, very complicated. You just need a source of electrons, you need just a couple of electromagnets, you just need a detector, so it might be like, I don't know, 200,000 euro. But modern electron systems, they are sometimes above 1 million euro in price, which is actually because you need to precisely control. Well, good question, but I don't think we have time to answer it in detail. Basically, the emission rate of electrons can be controlled by many different values. If you use a semiconductor as a source, you can vary temperature, you can use special grids that block some electrons, dipole, quadrupole systems. There are lots of interesting technical solutions. Sorry for not answering your question in detail, but really it will take too much time. So we can talk about this maybe after that, if you like. So now I think we are ready with this point, and so that we can go ahead with our main topic today, which will be the power of fluorescent microscopy. I want to convince you in a logical way that fluorescence microscopy is not just kind of like extension of bright field microscopy. It represents a totally other concept of generating images, which brings lots of advantages with it. This part about phase contrast, I will skip for a while. Maybe later we come back to it, because you know, in your script, the logical flow in the script is sometimes different than logical flow in the lecture, okay? So let me convince you that fluorescence microscopy is better. First reason I want to bring to your attention is, imagine you are in the room full of glowing or full of light. There are billions of lamps and they are all producing light. And now somebody tells you, look, your task is to find one single lamp that does not produce light. And I say, oh, wow, that's hard. I have so many lamps and they're all glowing, and now I'm searching the only one that is not producing light. It is difficult, right? I think you can imagine this. This is exactly the situation you have in the light microscopy, because you have basically lots of light coming from your light source. Many objects in your sample will let the light through, and there will be only a couple of them that you stained, that you are interested in, that will not light go through, and they will appear as darker objects. But even psychologically, it's hard to find them, harder to see them, okay? Especially if they're small. And now we have another picture, and I think it's very easy to see. If you have a dark room filled with billions of lamps, but they're all not glowing, all not emitting light, and only one single lamp produces light, and say, sure, I see it immediately. And this is again exactly the idea of the fluorescence microscopy, because your whole background is idle, it's dark. And the point that is interesting for you, it can be a virus particle or even a single protein, it emits light, and this is what makes you seeing it immediately, even if it's very small. Even it's like a star on the sky, having really very small angular dimensions, still you will see it. You will see it quite clearly every time. So this is first what I wanted to illustrate to you. Another advantage that I think is also quite easy to understand, and I'm talking about principal advantages of fluorescence microscopy, is that let's compare now again bright field classical optical microscopy. You have a light source and you let this light to go through your sample, and in the end you see some picture, and the objects that are interesting for you, they absorb light, they are absorbers, and you can make them even more absorbing if you stain them with some dye, like with crystal violet or with trypan blue or whatever. But look, in fact, they absorb it, but you have no way to amplify this signal, because it's sort of absorption. You cannot really amplify it. It can be more absorbing or less absorbing, but there is not much freedom in your hands, because absorption is something that you cannot really amplify from your equipment, because it's sort of absence of signal, and you cannot really amplify absence of something. Okay? And now again, let's change our field and enter the field of fluorescent microscopy. In this case, the molecule emits light. The object you are interested in, it produces photons, it produces energy. And if you have signal and idle background, you can amplify it. You have some maybe very simple, very little signal, but now you have lots of opportunities to increase it. And one of them is shown here in the system that many of you maybe already know. It is a photoelectron multiplier. So I have one single photon that enters this amplification system. It hits the target, and here one or two electrons will set free, maybe one at the beginning. Then this electron will be accelerated by very strong electric fields as it goes from this point to this point. Here it hits the target again, but this time it has so much kinetic energy that it hits two electrons, and now two electrons will be accelerated to the next target where they hit it, and they now have a lot of kinetic energy to emit more and more and more electrons, so that in the end you have 10,000 times more electrons. And then in the output from one single photon you get 10,000s of electrons, and when you convert them back to photons, which is quite easy actually, you have a lot of signal, and this is what I tried to show here. Okay, So maybe not so obvious at the beginning. I hope that you saw that these are logical and fundamental differences between the standard microscopy and the fluorescent microscopy. And so now we can appreciate why it is so, popular in cell biology and in medicine. So this was our second point, power of fluorescent microscopy. If you know some more arguments, you're always welcome to comment live or in chat window or later, whatever you like. Now we will move towards phenomena called Jablonski diagram and Stokes shift, and we will talk about some features that are used in a modern fluorescent systems. So this will be the goal of the remaining part of this lecture. Again, I want to apologize for giving maybe some informations that you already know. The problem I have to handle here is that your group is very heterogeneous, and I know it. Some of you I know well, some of you are new, but what I definitely know is that you all have different background, some more medical, more biological, whereas some others more engineering background. I have to address both backgrounds somehow. That's why let me repeat some basic things, and I hope it will not disturb you too much. Our next question, which is exam related, is a Jablonski diagram, or sometimes called Jablonski diagram, but this L is actually this uh, Polish, so the special letter, which is also used in former currency, zloty or Jablonski. So it's pronounced as U actually. So Jablonski diagram shows the energy distribution in an atom or in a molecule where we have just actually single axis. This is the y-axis from up to down, and it shows the energy levels in your electron system or in your atom. So here we can see so-called S1 level. This is the background or ground level. And this is the level electrons have in a molecule which is not excited. So this is a basic level. This is a molecule that is doing nothing, which is totally idle at the moment. And as we know, physics requires that any system always finds the minimal possible or minimal available energy level. And this is what corresponds to these levels. Some of you who remember chemistry, you may think about those facts like electron shells are filling up, one S1, one S2, one 2S1, 2S2, p orbital, next s orbital, next p orbital, next d orbital, and so on, right? So this energy filling in chemical elements. And what happens actually at the moment we have photon interacting with matter is that photon can, what we call it, like promote an electron to a higher energy level. Now electron is still part of the molecule, but it changed its energy state in a jump. Quantum physics says that there are no intermediate steps here. So it can be either everything or nothing. So it's just, you cannot just jump here. If you get enough energy, then you reach this level. If you have not enough energy, you just remain on your initial step. So it's really like a everything or nothing response because energy is quantized. And Now we are on next levels, S1 and S2, and the event that we call here is what we see here is called absorbance or absorption, right? So this is the moment where electron absorbs energy of a photon, and due to this additional energy, it is promoted now to the higher energy level. And a couple of nanoseconds later, usually it takes really nanoseconds in scale, this excessive energy, this additional energy will be given back by your electron, so it gets back to its initial level, and by doing so it emits light, and this is what we call emission, and this is the moment our sample produces light. If it is a delayed event, like shown in the next slide, if you have first absorbance and then a couple of minutes later or even hours later it again emits light, this delayed fluorescence is called phosphorescence. And this is phenomena, again, most of you are familiar with. Lots of modern toys, some, watches, many indicators, they use this phosphorescence. When you expose something to light, in the darkness, it will produce light with delay. And this delay is possible due to some peculiarities of electronic system. It has some features to stabilize excited electrons. It has features to store energy. And just few elements of few molecules are capable for storing energy and then emitting it as light later. One of them is phosphorus, and you know that's why this element phosphorus is called phosphorus. It means producing light, right, or um, illuminating. Okay, so the question was what Jablonski diagram is, and this was this way of representing energy transformations in a molecule or in an atom. So schematic showing these processes. Next point we have to discuss briefly is so-called Stokes shift. It's also quite easy and I should do it quite quickly. Stokes shift is related to the fact that the light emitted by your sample after you excited it is always shifted towards red region. It's always shifted towards longer wavelengths. It's sometimes called redshift, but strictly speaking, we better call it long wave shift. Because if you excite at red, your system will produce infrared. So it will always be shifted towards longer wavelengths. The reason is for this is quite simple, actually. And this is the fact that, let me come back to Jablonski diagram. This is just due to the fact that as you excite your system, you put some energy into it. And although the emission follows excitation within nanoseconds, still it is enough time for this electron to lose part of its energy by some other ways. By giving it as vibrational interactions with other atoms and molecules. And so it lost some energy before it has emitted it. And so that means that the energy you get back now as emission is less than the energy you've given before. And due to this, we have shorter wavelength in excitation, and emission will have always longer wavelength, which corresponds to lower photon energy. And this is nicely visible here in, if we just check the absorption spectrum of some hypothetic compounds, and if we check the emission spectrum of this compound, we always see there is a shift towards red region, towards long wave. And this difference between the peaks we call Stokes shift. It can be large or small depending on temperature, depending on which compound you use, depending on which wavelength you use, depending on many factors, in fact. But this shift is always present in classical fluorescence. It has some important practical consequence because it means always that you need to use one sort of light or one wavelength to excite your sample, and it should be relatively short wavelength. But the wavelength you are detecting, it must be different from your excitation wavelength. So you need to take care that you do not get this excitation wave into your detector. You need to cut it off. You need to filter it out so that you are going to detect this wavelength, not this one. So it makes some interesting technical solutions possible. So let me show just how different the Stokes shift is for different compounds. Here we see some common fluorophores that are used in modern fluorescence microscopy. Some of them you might have heard before, like FITS, like eosine, like Cy3, which is used in microarray DNA microRNA analysis. What you may also heard about phalloidine stained with FITS, or rhodamine, or Texas red. There are lots of them. Part of them just shown here just to illustrate how different these Stokes shifts are. This line represents that for this Lucifer yellow, the Stokes shift is large. That means your wavelengths are very different from each other, and it's easy for you to distinguish between them. So basically you use blue light to excite your sample, and what you get back is yellow light, and you can clearly cut them, and you do not mix them. In some cases it's going to be more tricky if your compound, for example this Cy3, if you excite it with green light 550 nanometer, and what it emits is also green light, but not 550 but 560, and then it will be a bit tricky for you to separate this 550 and only to get 560 through, because the difference is really minimal. For example, human eye cannot see this clearly. For you, both these waves will appear just green. You will not differentiate, but your optical system can. Here I think it will also be suitable time to talk about common light sources used in fluorescence microscopy. Initially, fluorescence microscopy historically used some mercury lamp, which produces, well, not such a nice spectrum. So it has lots of lines in the spectrum, lots of bands, and was not perfect, but was okay for the beginning. But then later they have discovered that usage of such mercury lamp or even xenon lamp is not so good, because they consume a lot of energy, they produce a lot of light, and most of this light is somewhat not used efficiently. Moreover, as this light goes from the lamp into your optical system, you lose a lot of it, so that final light intensity is quite low and not very convincing. And the revolution in the illumination in microscopy has been made with start of usage of laser systems, of course. So nowadays, I would say all systems, what I know at least, they all use laser beam, laser light source to illuminate the sample. The advantage of it is, of course, perfectly clearly controlled wavelength, extremely high intensity, and the option that you just scan through your sample by using the laser beam. You can make it so thin that it will address your sample like pixel by pixel, point by point, and you can really scan it by using laser beam, which is a lot of opens lots of new interesting options. On the other hand, laser light sources, and here you see some of them that used in a modern microscopy, argon-based system, helium neon laser, krypton argon laser. They have two disadvantages, in my opinion. First disadvantage is that, well, once you have one laser system, mostly it uses one mode of laser production, which produces one single wavelength. And they'll say, okay, but I need another wavelength. And laser says, sorry, I can only produce this wavelength. If you want to use another wavelength, you need to apply another laser. You need to have another laser tube, which is of course very expensive and not so nice, right? So laser is very specialized, can mostly produce one wavelength only. Another disadvantage is that due to its high intensity, laser light, if you do not care, if you do not pay attention, it will really destroy your sample. It is so intensive, it's so strong that once you let it go through your sample for a couple of minutes without moving it away or without switching it off, uh, you just lose your sample. It's just destroyed. It just evaporated basically. So, and again, those are factors that make modern fluorescence microscopy systems quite expensive. 2 weeks ago, I was writing a review, Gutachten, for DFG, for German Research Society, where I needed to evaluate some research proposal. And in this research proposal, professors from Stuttgart, they wanted to buy a microscope system. And nowadays, most universities and most like Fachhochschulen, like our, just cannot afford such a price because the system they wanted to buy was about, I think, 600,000 euro. 600,000 euro for a microscope. Quite a lot, right? So that's why nowadays so expensive they are. By the way, together with Mr. Porst, we also want, meanwhile, to improve our microscopic system in the lab. We use quite an old one, confocal laser microscopy, but we want to upgrade it. And again, I think for that we will require lots of money because of all these issues. You need laser, you need lots of different lasers, you need a lot of additional setups and equipments in order to produce high quality images. Well, let me summarize. I tried to overview the diversity of fluorophores you use for fluorescence microscopy. There are about 500 of them, meanwhile. Part of them we see here as a list. Those are fluorophores. These are compounds we use to stain our sample. And nowadays, you can find suitable compound for any purpose. If you want something blue, you find something blue. If you want something yellow, you find something yellow. If you want something blue that should be excited at UV and emits blue, no problem. If you need something that should be excited at UV light and produces green, no problem. You have nowadays lots of fluorophores that are totally suitable for you. Second, what we've done, I tried to explain this issue related to light sources. Mercury lamps are not used anymore in such systems, just in the cheapest probably, but mostly we have a set of different laser tubes. And every laser tube can produce certain wavelength. And here you see these lines here. Basically, you see which laser sources are better compatible with which fluorophore. So in all cases, you need to check both. Depending on the laser tube, you have certain choice, certain group of fluorophores you can use to uh, apply for microscopy. Why do we have so many different fluorophores nowadays? Why it is so versatile, so flexible? Well, the reason is simply that you can modify the color quite easily chemically. Sometimes you just change one single group in your molecule and it changes its properties totally. So here's a good example. Two molecules, there are slight differences between them here, side groups here, side group here, and they have totally different spectral characteristics. So one is red, another one is green, and this is due to the length of the pi conjugated system, which we discussed in our Biophysics 1 course with some of you already. Okay, now the only thing that is left to us to discuss is the main components and the procedure in laser scanning fluorescence microscopy. So just the main components and the main principle of working, of operation. So here we have the setup and you see already some parts of our system. First of all, we need to take care that we use just certain light wave, certain wavelength, which is suitable for your fluorophore, which is suitable for your sample. And in this case, it will be blue. So we need, if we have like complex light source, we need to put a filter here to make sure that only this blue component goes there, because all other components, green and red, they will not bring anything to image, but they rather bring additional energy, which will destroy sample. So here you try to be careful to minimize the amount of energy you bring to the sample, because otherwise you can destroy it. So here you try to be very specific. If you use just a laser beam here and it only can emit blue, then you are lucky, then you don't need this filter here. You just simply let this laser beam through. Here it will be then reflected by a special mirror, which is called dichroic mirror. And we will see now why it is called dichroic. Just comes in a couple of minutes. So this dichroic mirror reflects it, and now our sample is illuminated by this blue light. Couple of nanoseconds later, shifted due to Stokes shift, the light emitted will be produced by the system, will be emitted, and will be again collected by your optical system and goes backwards. And this time we see why this mirror is called dichroic mirror, because in fact it sort of reflects, but at the same time it allows some part of radiation to go through. So it has some dual nature. So it's a mirror, but at the same time it's semi-transparent mirror. So it lets emitted light, green light, to go through, and here the place where it needs to be detected. Also here we will probably require one more filter. The reason is that the light coming from the sample is contaminated by lots of additional light sources. It contains some blue components from the excitation. It contains some secondary harmonics. And actually, what we need to have is one single wavelength. So it needs to be selected from the rest, and you cut the rest off so that only one component, one important wavelength remains. And this is the image you see. Not so hard actually from understanding. What we see here is called epifluorescence microscopy. It's called epifluorescence because the light which excites comes from the top, and also and then it comes back from the same through the same optical system. So epi means from the top. So light from the top, and the image is formed also upwards. There are some fluorescence systems that work a little bit differently. In this case, the light is situated beneath the sample. It illuminates the sample this way from the bottom. It goes through it, it excites emission here, and the emission is collected in your upper optical system. But such optical systems are not so nice. First of all, you have a lot of contamination with blue light. Second, it makes the system bulky. It makes it too big. So this epifluorescence is more practical, more useful, more convenient to use. So that's why most of modern microscopes use this epifluorescence principle. Um, Let me now show briefly what kind of filters and mirrors we use. Uh, and I think it's also quite fantastic we, what kind of options we have now. There's a called short pass filter. So short pass filter basically is used for excitation. It takes care that no long wavelength radiation reaches your sample because you want to excite your sample. You want only short wave radiation to go through. So mostly then use short pass filter. The profile of its transmittance looks like this. Very interesting, very strong cutoff boundary here. In the opposite, let me come back. So this was this here, short pass. In the opposite, as you are talking about detection filter, here you need to get rid of short wave components. And in this case, you use the long pass filter. And actually part of this function can be performed by this dichromatic mirror because it may also have some filtering properties like shown here. Interesting is also the fact that you can combine two filters, which opens yet more opportunities. If you take short pass filter and long pass filter, if you place them on the top of each other like this, you have a chance of getting one quite fancy system which only will let very narrow range of radiation to go through, to pass through, like shown here. And this is also one interesting feature you may require in microscopy, to combine different filters to make them work synergetically, and then you can get them producing such a very nice, very neat pic, which will go through. In the lab, we have lots of such filters. Actually, we play with them in our practical for optometry students. They love them, they apply them a lot. And just maybe it will be interesting for you, some of you, such filters nowadays are used in many sunglasses, in many glasses. In fact, it was shown that many people who have headache or migraine or some neurological disorders, maybe depression, they can stay more healthy or can improve their health if they use special lenses, special, in their glasses, which filter certain portion of light. So that's why nowadays in optometry, in the optics that produces glasses, Such filters are extremely popular. And for example, one modern trend is to filter the blue part of radiation and UV part of radiation. So for example, my glasses here, they contain UV filter. It does not let UV light to reach your eye. And it is believed that due to this, the cataract development can be inhibited. And also the blue part, there are also lots of blue blocker glasses. Maybe you even have such of them. They block the blue part because it's also very energetic. And it is believed that this is good for your eyes to not to get too much blue light. Honestly, it is not very true. We people need all parts of the spectrum. We need UV light, we need blue, and it all needs to come to our eye. So that's why basically filtering it by using some lenses or filters is not a good idea. But there are special cases. For example, if you use lots of digital devices that produce a lot of blue light, actually, then it would be a good idea to have such glasses, because then you can block part of this extremely blue light coming from the screen, which can improve the state of your eyes. And the other example is if you have some high sensitivity to blue or UV light, if you have some hereditary predisposition, then it makes sense. Then you may think about getting some glasses that would block blue and UV light and can improve health of your eyes. You can think about this, but just my important message is if you are healthy and if you don't spend 8 hours per day, like me, sitting in front of the monitor, then you basically do not need such filters, because our body requires the total spectrum of electromagnetic radiation. And if you do not get it, it can influence your health. There were lots of experiments performed with animals. If you have home animals, if you do not let them get UV light or blue light, they cannot proliferate. They feel bad and they die quite soon, just because you use wrong light source, just because they don't get enough sunlight. Due to this absence of couple of wavelengths, some animals can die in your, some pets can die. There are lots of experiments performed here, and maybe if you have some pets, if you have some animals, please take care that they necessarily get enough light. And it should be actually light of highest quality, sunlight. And that's mostly all what I planned for today. Let me summarize and finalize, and then we will have several minutes for your questions and comments. Here is what we talked about today. We talked about the main components of fluorescence microscopy. So here we have some epifluorescence system, which puts some light, it goes through, it reaches the sample, and then it's emitted and goes backwards where it is detected by the camera. Modern cameras are quite great as well. There are CMOS cameras, CCD cameras, and they are really great, by the way. And here you can control it additionally by using your eyes. So it will be like live checking. So you can check, and you can also make collection by camera at the same time. We talked about the light sources used. There are different sorts of lasers. We talked about what are filters doing there, low pass filters, high pass filters, dichroic mirrors. And I also tried to describe why and how we have different fluorophore types. Let me check if we discussed everything what I planned. The oil, power, common fluorophores, common light sources, common filters, Stokes shift, Jablonski diagram, band filter, pass filter, dichroic mirror. Well, in my opinion, we actually covered all the points that I planned for today. Now, I want to invite you to ask something, or if you need to clarify something, or maybe you can comment something. Maybe you want to ask to talk about um, sunlight or something. So just go ahead. This story with sunlight, by the way, there are several books devoted to it. I didn't know how important the sunlight is for our eyes before I read several books, and they are really striking. They are absolutely shocking, actually. There was, for example, a story about a man who he was like 60 and he had lots of troubles with his um, mm, joints. Actually, his joints, they were all ill, and most of doctors, they also said, yeah, you just need to go, you need to get implants, so your joints are just dead. And someday this man lost his glasses. They were broken, and he needed to wait until in the optics they will make a new glasses. And these 2 3 days he just spent outside. There was a good weather, and he spent this time without glasses. And suddenly, before he was getting his new glasses repaired back, he noticed that his joints, they were getting better. And he said, wow, coincidence? Well, let me consider, let me continue. And he was just continuing, not wearing glasses, like sitting, letting his eyes getting a lot of sunlight. And interestingly, as he did it 1 month long, he came to the doctor, and doctor just made such a eyes. They said it cannot be possible. Your joints are fine again. And such cases are known. It is actually familiar. Also, if you have like fishes in your aquarium, or if you have like guinea pigs or hamsters or rabbits or hens, if you do not let them absorb sunlight, they will they cannot proliferate. They will get non-fertile. It's just not possible. And the same, by the way, is true for human beings. It is also a known phenomenon that people who spend several months just without sunlight, just having artificial illumination, they start getting infertile. They cannot get children anymore. This is a phenomenon that may be partially responsible for this phenomenon observed in last years, that the number of births is decreasing, especially in the so-called developed countries. One of the reasons for that, one hidden reason, is that we use lots of artificial light sources and we neglect the influence of the sun. So yes, so some comments arriving, so let me check them. Yes, so one aspect what you mentioned was, of course, related to vitamins D production. We know it comes mostly will be produced by the due to skin exposure to UVB light, but it's just part of the story, because we know that vitamin D production is very inertial. So mostly you only experience beneficial effects 2 3 weeks later after the exposure. Uh, there are definitely many ways how sunlight acts on our hormonal systems, uh, but we only know this serotonin and melatonin system, which is responsible for our mood. But there are lots of more systems which we do not know about. Uh, several years ago, they discovered this intrinsic photosensitive neurons in ganglion cells in our retina, and that was, wow, revolution. But every month, actually, they are discovering new and new photosensitive cells in our ganglion cell layer in the retina, and they are all connected to different regions of the brain, hypothalamus, some behavioral cortex, and so on. That means there are lots of things that we still do not know about how different spectral regions of light have impact on certain regions of the brain. It needs to be studied. The phenomenology, but suggests strongly that if you do not care about, if you do not take care about it, if you neglect this light story, if you use wrong light sources, it may be very bad for your health. And by the way, there is an interesting theory that even a number of oncological cancerous diseases rises in last years just because people use wrong light sources. And that may be true because animal models show this trend. If you have animals and if you apply wrong light sources to them, they develop cancer more readily than animals that experience sunlight. Again, but we know overexposure to sunlight is also deathful because it can produce skin cancer. So that's why, as always, some gold middle must be met. Yes, can no longer reproduce. Yes. It also happens with horses. There are lots of studies with foxes done, and with many animals. You're right. So it's. But what I'm just wondering about, we have no such lots of such studies made for animals, and all people who have snakes or lizards at home, they always know that they should buy UV lamp for them because otherwise they cannot proliferate. But why don't we think about humans? And it's no mystery, actually. We humans also need UV light. It needs to be there. Without UV light, we are not really proliferating efficiently. Uh, yeah, it's just like this. So, what's the role of mercury in the microscopy? Um, yeah, actually, I mentioned just the mercury lamp that was actually a sort of very useful light source. And the mercury lamp, mercury as element has several electronic transitions from upper level to lower level. And each of these transitions produces certain wavelength, certain photons. Mercury luckily has several electronic transitions which produces like more rich spectrum, and it produces light of relatively good quality. And mercury was also an element which is easy to be excited by electric field so that it can start emitting light. So that's why modern it's called I don't know the English term, but Leuchtstoffröhre. So these lamps, daylight lamps we use, they have mercury inside because it's just useful. It emits light in a suitable range, and the fluorophore, which covers the daylight lamp, re-emits, so it absorbs UV and re-emits light, which is more comfortable for human eye. So mercury is quite useful, but we know it's not very friendly environmentally. That's why we have this trend to use LEDs nowadays because they are more environmentally friendly. Yes, so as one more point appeared, is there any effect of lack of light for people who live in countries their sunlight is short periods of time? Of course, definitely, it's absolutely known that if we just take into account the depression periods, schizophrenia, cases, well, just depression. There is a clear trend if you take a correlation between how much is the sunlight exposure of this particular country and depression. There is a very strong correlation between these 2 points. So the more in northern countries, the depression level is always higher. That's absolutely known. I saw the World Health Organization data, and they suggested quite strongly. Yes, and this is what you may also experience. For example, if you are like me, coming from some lands, from some country where you were exposed, uh, been exposed a lot to sunlight, and then, for example, Germany, it's significantly less. It's quite normal and expected that you will experience periods of depression and bad mood and what they call this, that you are tired, this. And in fact, the only way or the best way to get rid of it, to fight it, is to expose your eyes to sunlight as much as possible, or you use some good high quality light source like a lamp, and you let this light to enter your eyes. There are lots of lamps nowadays used for light therapy, and it helps. I should tell you, I'm using a UV light. I had the lamp here somewhere. Oh, I know, brought it away because it's almost summer now. But in winter time, I'm using just UV light, a black light lamp, and it helps me to stay more or less not depressed, because without such light support, I feel also sort of sick and tired in winter time. And maybe, who knows, maybe you feel similar. Partially, it is because of this. Okay, good. Then I think we, as you saw probably today, it wasn't very much and it wasn't hard. And this is approximately how I would like to keep it. Our lectures, to make it not very, overloading you with material. And I hope it was not stressy for you today. And we also discussed about some interesting things today. Thanks a lot for your attention. So now we will close this class and we see us next time. Uh, so many thanks. Have a nice day and see you next time then. Goodbye.